So good morning. Welcome to the second day of this coronavirus meeting. So we'll be sharing a session today. Des and Rachel is somewhere. Hopefully. We just popped out for a second. The session is moving forward onto wildlife coronaviruses this morning. And our first speaker is Marike Geldenhuys from the University of Pretoria. Thank you, Marike.
Good morning, everyone. My name is Marike Geldenhuys, and I'm a postdoctoral researcher at the University of Pretoria in South Africa. I'm actually a stand-in today for Professor Wanda Makotter, who sent me in her stead. She really wanted to attend, but she just couldn't get around to doing so. I didn't think I moved that. She sent me in her stead because I've been doing the coronavirus research of our group for the last 14 years. So thank you again for the opportunity to be her stand-in. She is the director for the Center of Viral Zoonosis at the University of Pretoria. We are in the medical faculty, and she holds 2 South African research chairs, which are funded by the Department of Science and Innovation, one in zoonosis and the other one essentially in One Health. And she's also co-chair for the One Health high-level expert panel of the quadripartite. So we are the Center for Viral Zoonosis, but our specific research group is the Biosurveillance and Ecology of Emerging Zoonosis Research Group. So we do interdisciplinary, transdisciplinary, interfaculty research with a One Health approach. So these are just a few of our team members. Currently, we have 3 postdocs. The postdocs are all in charge or coordinating at different virus groups like coronaviruses, paramyxoviruses, lyssaviruses, and then we all collectively take care of the filoviruses. We currently only have 3 PhD students, 2 MSc, and 6 staff members, but we do have certain years we've got a large research group, and other years, like this year, it's actually quite small for us. So we have excellent collaborations and partnerships and very, very good funding. Some of our funders include the CDC in Atlanta. We also have excellent funding from DTRA, the Defense Threat Reduction Agency. Nationally, we have the National Research Foundation and then the Poliomyelitis Research Foundation. So they really enable us to develop these strong partnerships that we have in governmental departments in South Africa, as well as museums like the Ditsong Museum in Pretoria. And then we collaborate very strongly with the National Institute for Communicable Diseases, which I think has the only BSL-4 in Africa. And then we also collaborate with institutes and universities in other Southern African countries like Zimbabwe, Eswatini, and Mozambique. So our focus is really zoonotic virus surveillance in small mammal hosts like bats and rodents, with the focus of doing surveillance and building diagnostic capacity, looking at the host pathogen interactions themselves, looking where animals and humans are interacting to see which kinds of activities increase or even decrease risk, and then looking at the ecology of the bats to be able to understand how these viruses or why these viruses occur in these animals. So our main focus is bats, but we've also looked at other small mammals. So this really is to understand what pathogen diversity is circulating in these wildlife species that have been identified as natural hosts for pathogens that could cause spillover, looking at where there are interactions between these natural hosts and other animal species, and then how both of these groups of animals actually interact with people, which is largely in a very negative and destructive way because of deforestation, agricultural expansion, that is increasing stress on the animals as well. So we aim to apply what we learn at a local level for conservation and improved coexistence between people and the natural world, and also being able to adapt the same lessons we have learned in one ecosystem to similar ecosystems. So we do a lot of field work in South Africa, but mostly in the northern parts of South Africa, though we have also done some work along the coast. So we may mainly look at cave sites because it's actually just easier to catch bats from caves. And the cave sites are also interesting interfaces where there is human activity. In Africa, depending on the culture in the area, we have caves where people go in for prayers and rituals. They sometimes bring animals from wherever to slaughter the animals in the cave. So we've seen chickens running around. And then some of the caves are also used for like ritual circumcision. So it depends on the community and the duration, but there's definitely that contact interface that people are entering these caves for several hours. So we catch the bats, we collect a certain number of samples from them. We also tag them so we can identify them again if we capture them later on with tattoos, as well as pit tags like you would find for dogs or cats. Just much smaller, and then we take morphological measurements so that we can, characterize the demographics of the bats that we have captured. We also have, some surveillance sites in other South African countries where we go to help them do field work, such as in Zimbabwe and Mozambique that are starting later this year. We were just recently in Botswana at the university there to help them plan new projects and develop their field work. So just a quick reminder of coronavirus taxonomy, but coming from the bat angle. Of the 14 alpha coronavirus subgenera, 9 are actually associated with bats. That includes the 2 human subgenera, the Duvina and the Citricoviruses. Amongst the beta coronaviruses, of which there are 5 subgenera, all except for 1 is associated with bat coronaviruses, and this is the Mbecovirus, which contains human, bovine, and rodent coronaviruses. So these ones that include bats, of course, are the severe acute respiratory syndrome-related coronavirus species in the Sarbecovirus genus, and then similarly the Middle East respiratory syndrome-related coronavirus in the Mirbicovirus subgenus. So these other 2 are exclusively associated with bats. One important thing to just remember about bat coronaviruses is that they are genetically diverse and they're globally spread out. There are also known coronaviruses in bats that are very related or highly similar to coronavirus species in other animals, and there is a suggestion of a history of crossing the species barrier. One last thing is that bat coronaviruses display a level of genus host specificity. So you'll find related coronaviruses in host species within the same genus. So to do surveillance, you really want to have an idea of what was done before you just jump right into it. So we did a literature survey in 2021 and have been updating our literature every time something new is published. So bat coronavirus surveillance essentially began in 2009 on the African continent. I'm only speaking about Africa now. There's 26 publications that have reported bat coronaviruses from various countries. And this accounts to just over 27,500 bats being tested, which seems like quite a lot. Most of this was cross-sectional research that has been done, where they report on virus diversity as well as expanding host diversity. So from this, we know a lot about what diversity is there and which hosts you can expect them in, but we understand very little about the maintenance of these viruses and how or why they are being circulated in these populations. There is also a level of generalization across all bat coronaviruses, detected in different hosts, or different types of viruses, but yet they're usually collectively generalized, and it is not yet clear if that is a good idea to do or not. So there's also very limited genetic descriptions of these viruses, as they are mostly available on online databases as very short sequence regions and very few full genomes. So essentially, in a nutshell, what has been found is bat coronaviruses related to each one of the human subgenera, except again, of course, for the Mbecoviruses. So there's a large diversity. There's also lots of unclassified, uncharacterized viruses. So instead of this presence-absence detection of viruses, we strongly advocate for more longitudinal studies that look at this whole scenario in a more holistic way. So to look at the viruses in the bats themselves, look at them in the populations, how and why these viruses are being maintained, when is there potential increases of excretion from the bat populations, and is there in contact interfaces between bat populations and animals in the area. Of course, there are definitely animals near bat roosts, and they likely have their very own cohort of coronaviruses as well, which nobody is really looking into. You can also even consider these animals as potential mixing vessels for recombination between potentially spillover bat coronaviruses and their own repertoire of coronaviruses. And then, of course, lastly, you have to look at human activities in the area that might be doing things that could be increasing or even decreasing the risk of spillover by causing additional stress on the animals. So all of this allows you to build your picture of what is circulating, what's the timing of the circulation, what is the risk, and then maybe being able to apply small interventions to decrease the risk of spillover and the emergence of new viruses. So we applied this whole approach to a maternity colony of this fruit bat species, the Egyptian rosette bat, or Rosetus aegyptiacus, which is also known to host a multitude of other viral pathogens like Marburg, for example. So this is a maternity colony. It is surrounded by a nature reserve on one side, agricultural lands on the other, and a human settlement throughout, so quite a rural landscape. So we collect fecal samples from these bats on a monthly basis. We've done this since 2017, and this is still ongoing as well, but we've looked at the first 2 years of data. So we collect rectal swabs from bats that we capture from harp traps. We also take their morphological measurements, so we know their sex, their age, their reproductive status, so we've got that demographic data. And then we collect fecal from inside the cave, and this is predominantly just to expand our sample volume and our sample size, because we get very limited material from rectal swabs, so it actually makes characterization of the viruses very challenging, because remember, this is often very low concentration positives that we get. So we test them for nucleic acids, we analyze the data with statistical models. We also characterize some of the full genomes using an amplicon-based strategy as well as unbiased sequencing. And we detected just over 200 positives from the 1,400 samples that we got. So there's a comparative level of positivity amongst the fecal and the rectal swabs, though if you account for pooling, it's much lower in the fecal samples. We actually detected 3 different lineages of coronaviruses. Sorry for the very, very small text, but the colors will help me explain this. So we identified an alpha coronavirus that was very different to anything that has been described before, so it likely represents a new species. The majority of the positives were these red sequences that is related to a bat coronavirus called HKU9, which I'll explain in a minute. And then the last beta coronavirus was very rarely detected and is not the same as this HKU9 diversity, as they only share about 72% identity in this very short region. So nobicoviruses, or this HKU9 diversity, HKU9 is a species within the nobicovirus subgenus, actually occur in fruit bats wherever all wild fruit bats occur, Asia, Africa, Australia. So you find these viruses to be very widespread. They're also very abundant in these populations with an average prevalence of about 7%. These can also be very large colonies, and throughout different parts of Africa, there are areas in urban cities where fruit bats also occur in these cities. So there is definitely that contact interface between bats and people. Even amongst cave roosting bats, like the Egyptian rosette bat, they also need to leave and find fruiting trees to forage at, which often gives them into contact with people and other animals. These viruses have also are genetically quite diverse. They have lots of strains and variations. One of the species in Asia actually picked up a gene from a different virus group entirely. So there is that level of recombination that is occurring. And those viruses have actually, or the nobicoviruses have been identified by several independent research groups, as well as online spillover risk ranking tools, as viruses to watch for the possible emergence of as new novel agents. So I don't want to get into too much detail, but we characterize some of these viruses. We're still busy with the alpha coronavirus because it's so different to anything that has been described. It makes its characterization a bit challenging, but we've recovered about 68% of the genome. We also used an amplicon-based deep sequencing approach to recover 4 strains of the HKU9 diversity. And then when we compare that to the full genome of this novel beta coronavirus, they only share about 64% similarity overall. So this is not the same as this HKU9 diversity and will likely represent a new species when we describe it further. We would love to actually sequence every single positive and get lots of genomic data, but that's not financially feasible. So we decided to look at the spike genes of this HKU9 diversity. And very interestingly, we thought they would be much more similar to each other, but they're not. They share between 92 to 99% amino acid similarity. If you look at the phylogenetic tree, at certain times of the year, they do group together, but they do not all group as related to one species. If you look at some of these darker sequences in the text, those are from other African countries. These two are from the same host genus, but this one at the top is from a completely different host genus, so that there is an element of viral sharing between different bat genera. So we also try to monitor if we can see extinction or domination of some of these spike genes. But I don't think we have sequenced enough of them because they really are just all over so far. Then we also wanted to look just briefly at the receptor binding domain, and we actually saw between 0 to 16% amino acid differences within just this region. So this is very, very new data. We haven't been able to analyze it properly yet. So we need to interpret what this actually means, what it means for adaptation to new hosts, etc. So in terms of the ecology of how these viruses are being maintained, we see these excretion peaks at the start of the year. So this we did with a general linearized model as well as GAM models. We're still tweaking the models to be able to see if these models are correctly being interpreted. But we do see these clear excretion peaks at the start of each year with low-level excretion throughout the rest of the year. These peaks are also, the overall peak you see here is largely driven by these HKU9 diversity as well. So if we go back to what is happening at the population at the start of the year, this is actually when so how it works is rosettas have one breeding season throughout the year. They accumulate at this maternity colony in spring, in September. They have their pups in October, November, and between February and April, we've got these young weaning bats that are becoming independent and flying around. Towards May, most of the bats actually leave the colony to go to different roosting sites. And then throughout winter, there's a very small population that stays behind that generally don't look very good. They're fighting each other, for example, and then it starts all over again. So what we see happening here is that this is the influx of the potentially immunologically naive young bats that are picking up positives, right? So if we then look, sorry, everything is very blurry. If we then look at the demographics of the bats that were actually positive, we see that the overall excretion trends we see are in fact being driven by these young bats. And this is true for all of the lineages except the novel beta coronavirus because we had too few positives for strong statistical inferences. So, like I said, we also tattoo our bats to be able to monitor recaptures. And this might sound much easier than it actually is. We only have 11% recapture rate within this data set. So only 75 bats. Of those 75, only 19 were actually positive for coronaviruses at any given point. Of those 19, we could only use 10 to determine viral clearance because they were positive at their first capture, and then we could monitor that after that. So there was only one bat that was positive for coronaviruses in 2 consecutive months. First a beta and then an alpha. So I mean, this is one data point, but maybe it suggests that there isn't cross-protection, which also makes sense because these viruses are very different. And overall, it would suggest that viral clearance can occur within 4 weeks. But we really need to follow up this data with serological analysis. But this is at least a beginning to form a picture. So we are building our little picture here. We know what's going on. We know what's circulating. We can infer that it's probably young independent, immunologically naive bats that are losing their maternally derived antibodies, that are becoming infected by some of the bats that are remaining in the colony, that then drive this amplification peak. Next up is we will definitely looking at serological surveillance now that we've got some genomic data from the viruses to specifically be able to do serological surveillance of this bat virus diversity and expanding it into other animals and humans as well. So in terms of other animals, we also wanted to see what has been done in non-bat animal surveillance. So we looked at published literature as well as these predict online databases. And overall, about 32,500 animals have been screened for coronaviruses. This is just nucleic acids. And this seems like a lot, but if you consider also that this is across 15 countries, across many orders of animals, and very opportunistic. So this actually means that less than 1% of these animals were positive. So the positive came from rodents as well as chimpanzees. The chimpanzees were actually a reverse zoonosis event in, I think, the Ivory Coast. So animal handlers actually gave the chimpanzees, I think it was OC43. And then amongst the rodents, we know that they host their own repertoire of viruses. So this is not sustained longitudinal surveillance where similar populations are being surveyed. It's more opportunistic sampling. Then we also did some rodent sampling in South Africa where we detected a very low positivity, but rodent sampling is not easy. It's not like bats that you can capture them all from a net in front of the cave. It's very opportunistic. You catch whatever you can. And we also detected viruses, which was expected, but what was very interesting is that we also detected novel alpha coronaviruses that grouped in clades, which of course you can't see here, that grouped in clades with bat coronaviruses from the same location. So there's a suggestion of viral sharing that's happening between the bats and the rodents, and this is a very active field of research that we are busy working on. Then, in terms of controlled and notifiable diseases in South Africa, there are no coronaviruses on this list. So there isn't really incentive or a drive to do coronavirus surveillance generally in animals in South Africa. Unfortunately, I also tried to see if I could find any literature on this, and I couldn't. So I think if there is any general animal coronavirus surveillance going on in South Africa, it's probably in some vet lab on a database that's not being released. So we really plan on expanding our domestic and livestock surveillance in the future around these bat sites because we don't have the manpower to do bat and animal and human surveillance initiatives. We decided to let the bat research direct our next steps. So we will also be doing targeted serological surveillance based on the known bat viruses we've discovered. And this was just an example of one bat interface that we're looking at. There's multiple with different bat and coronavirus diversities as well. We collaborate with the Chris Broder Lab at the Uniformed Services University in the USA, and they can actually express viral antigens for us based on sequence information that we supply to them. We take these proteins and we couple them to beads that we use in Luminex assays that we can then multiplex and do multiple agents at the same time. So we also are planning on developing these assay tools and sharing it with our collaborators in different SADC countries. In light of this, we actually had a workshop that was funded by the UKHSA last year in April, where we invited representatives from different SADC countries to come and join our workshop, essentially. The aim of the workshop was to develop or assess gaps in research and see if there are a requirement or a need for surveillance plans in some of these other SADC countries. We also made them fill in a pre-workshop questionnaire where we could assess gaps and knowledge issues, essentially. So the whole aim of the workshop was really to establish collaborations and networks. We tried to invite members of the environmental sectors, the veterinary sectors, and public health from each country so that everybody had a voice. It was interesting to know that some people working in the same sphere with the same goals had no idea that other people were doing the same things. So in that sense, it was quite successful in developing these collaborations. And then we really wanted to assess what gaps there were and if there was a need for developing an in-country research plan. So essentially, the take-home message here is that, yes, there is a need. There is this gap, and there is an interest. The delegates knew that this is something that their country needed to expand on, but of course, like everywhere, funding was a problem, as well as capacity building. There was very limited expertise in various of these countries. So with our funders, we've decided to help wherever and however we can. So we have developed new collaborations, like recently with the University of Botswana, to help wherever we are capable, essentially. So without taking up too much time anymore, so thank you very much for the sponsorship for today from the Queen's University and the UKHSA. Thank you very much for listening to me, and please come talk to me afterwards, even if it's just suggestions or advice or anything. Thank you.
Thank you for a lovely talk. Do we have any questions from the audience?
Thanks, really nice talk. Surrounding the assays you're developing, how fine can they be to distinguish your different strains, as shown in your tree?
So the assays we have currently, we at first initially thought we're not going to do coronavirus surveillance because what was available was not specific enough. So we thought it's going to be very non-specific, so let's exclude it. So we've got some great tools for paramyxovirus surveillance and filoviruses. Then we decided we've now got this nice genetic information, so let's see what we can do. And I also advocated that we don't just use for example, Asian sequences of SARS-related viruses. Let's use the stuff that has been found in Africa because it's very different. So we decided not to use nuclear proteins because we thought that that would create chaos with some of the serological cross-reactivity. So we use full spikes as well as spikes based on the receptor binding domain. We haven't yet done this, so it will be a lot of trial and error. But I think using both for all the viruses would be able to maybe eliminate more non-specific binding if it's focused on the complete spike protein and then comparing it to just the receptor binding region. Yeah.
Hi, Marike, great talk. The novel alpha coronaviruses in the rodents you mentioned toward the end, is it possible they're leukocoviruses? I see leukocoviruses are on your chart, and leukocoviruses are a little odd because they're classified as alphas, but they really have like a beta coronavirus-like spike, so they're in some kind of intermediate territory between alpha and beta, and that sounded like what you were describing. So have you looked closely for leukocovirus homology there?
I think they are within the meninacovirus, the diversity that you find in Miniopterus bats. So I'm not sure how related they are to what viruses did you say?
They're called leukocoviruses, so JC34, and it was on one of your charts. So JC34 is the famous one, and that's the prototype. But they're very understudied, but actually possibly more important than people realize, actually, I think.
I think the diversity of these alphas are more related to the Miniopterus clades. I think it's Municacovirus, these subgenera. I don't know if I'm pronouncing that correctly. We also saw sharing between very different bat genera in that same clades. So we're thinking that those are just viruses that are being put out there, that are being detected by different, or picked up by different animals. We also don't know if those viruses were replicating or if it was just, a once-off detection in rodents.
Thank you.
Really great talk. Does your group or do any of your collaborators try to do virus isolation, or are you strictly doing molecular techniques right now?
We try, right? There's a lot to do, and we are quite a small group at the moment, but that is our dream. All the postdocs want to get back to the cell culture research, but it's just, we do all of this. So we do the field work, we do the lab work, we manage students. So that if you give me a month and say, do what you want, cell culture is definitely something we want to do. We have also in the past, we developed cell lines, but they were not necessarily the correct cell lines. It was more of a, can we develop cell lines proof of concept, which we did. Our idea is to make, for example, from rosettes, an intestinal cell line to be able to see if we can grow these hosts' viruses within their own cells, but then also pair it with like trypsin cleavage of the spikes that we know they need some extra help. But we haven't yet done that. It's something we really want to do, yeah.
I don't know much about bats, but I'd assume that bats in colonies stayed in that colony for their life, but I think you're suggesting there's quite a lot of swapping between colonies, is that right?
It depends on the species. Some bats, like Eidolon halvum, which I spoke about, they migrate from, I think it's as far north as Ghana, all the way down to Zambia. So really fast. I mean, anything that they have, they are spreading along the way. It's not one flight, one direction. It's like little pit stops all over. Some bats decide to stay in, you know, DRC, and the rest of the colony moves down. So it's really depending on the species. And we actually try to track bats to see where they're going. Depending on, again, the species, your tracker limits you of what you can actually put on a bat, because it has to be a certain weight. Ideally, also, if bats live in trees, you can use solar charged batteries, or bats live in caves, so that makes everything just much more difficult. So they can travel certain, up to 100 kilometers, even in an evening, just to forage. So they really do travel quite far.
They might move from one colony to and then live in different colonies.
Yes. So even in this particular cave site, we actually think that there's that cave and a sinkhole on top of the mountain. So sometimes the bats also just move between that. We actually think the caves were connected, but there must have been a rock slide or a collapse or something like that, because we can't find the connection. But we have seen bats in the sinkhole, and because we can't actually get down there, we don't know if it's the same bats, but this is what we are suggesting.
Okay, great. Thank you, Marike. Pleased to now introduce Richard Delahay from APHA in the UK.
Thanks, Darren. And thanks to the rest of the organising committee for inviting me to give this talk. I've got an admission to make to start off with. I'm not a virologist. I'm not a microbiologist. I'm not even a vet. I'm a wildlife biologist, and so my talk is going to take a little bit of an ecological perspective on things. So the work I'm going to describe is the result of a collaboration between the National Wildlife Management Centre at the Animal and Plant Health Agency and our colleagues in the virology group there. And I'm going to talk about some work that we started a little while back to look at SARS-CoV-2 infection in wild mammals in the UK, and principally I'm going to focus on European badgers. So a little bit of background, which no doubt you won't need anyway. During the pandemic, there were a growing number of cases detected in free-living wildlife. We're interested in free-living wildlife because of the potential for the emergence of persistent reservoirs of infection, and the possibility that spillover from humans into wildlife then results in spill back from wildlife into humans. And of course, if the pathogen can circulate in wildlife populations, there are opportunities for it to evolve into perhaps new variants with different characteristics. And that could be challenging if spill back to humans occurs for effective control of disease in humans. And with limited resources, we need to carefully think about how we prioritise in terms of surveillance and risk management in terms of wildlife. We can't sample everything that that's out there. And given such widespread infection in a human population of a zoonotic pathogen, then we really need to think about the implications of infection in wildlife. So one group that emerged as being of particular interest during the course of the pandemic were mustelids. The Mustelidae is a family of small to medium-sized carnivores, and it includes many mammals that you'll be familiar with: weasels, minks, otters, the many different species of martins. And also, importantly for this talk at least, the European badger. The European badger is a common, widespread mustelid in the UK and in Ireland. And experimental infection studies during the course of the pandemic demonstrated that mink and ferrets were susceptible, although the outcomes of infection in those two species were quite different. But shedding and transmission to in-contact animals was demonstrated in both. So we knew that mustelids were a group of some importance. And then along came some explosive outbreaks of infection on mink farms. Some mink farms in Europe, in North America and Southeast Asia were affected by SARS-CoV-2. And they demonstrated rapid and effective transmission amongst the animals. And also spill back into human populations. But what about infection in free-living mink? Well, there is some limited information. In North America, it was demonstrated that free-living mink captured in the vicinity of mink farms were infected. And also in Spain, mink that were not quite so close to mink farms, but still within the commuting distance we would expect of this mammal, were also infected. And we know from previous work on other diseases like Aleutian mink disease that you can detect hotspots of infection in free-living mink through identifying mink farms with infection, because free-living mink are attracted, generally speaking, to mink farms. So what about the potential for mustelids like mink to maintain infection in the wild? Well, of course, their ecology is very, very different. Their spatial organisation, their density and so on is very, very different in the wild to what it is on mink farms, where they're crammed into cages in very close proximity to one another. Because mink, just like most mustelids, are actually solitary. For most of the year, most of the time, they're solitary. So the typical pattern of mustelid social organisation is that you have the males with very large territories, and within their large territories, they will have smaller territories belonging to females. But the males and the females only come into contact infrequently during the breeding season. And because the males are territorial, they tend to stay distinct from one another. So there's very limited contact in the population amongst individual mink. Now keep that in mind, because I'll return to that broad theme a little later on. So we carried out some surveillance for SARS-CoV-2 in wild mammals in the UK, and we looked at a range of different species. When we first started out carrying out our work, we were carrying out opportunistic sampling, so we were really piggybacking on existing research programs, existing wildlife management activities like hunting and pest control and so on, people who would come into contact with animals and collect samples for us. We also reached out to rehabilitation centres, wildlife rescue centres and so on. And during the course of the pandemic, we identified certain priority groups of species that we were particularly interested in, and that changed during the course of the pandemic as more evidence emerged about infection in different species. But one species that became of particular interest, to me at least, was badgers. Now, the samples that we collected from badgers largely came from a long-term field study in the southwest of England. It's actually been running since the 1970s, where we've been catching badgers in these cage traps. We've been anaesthetising them and marking them, taking a whole bunch of samples. But this is principally for the detection of bovine tuberculosis. But it seemed like an excellent framework that we could use to capture information on SARS-CoV-2 infection. We also, as I said, reached out to rehabilitation centres. Unfortunately, we only managed to get one sample from an animal in a rehab centre. So the vast majority of the samples we have come from this long-term study in the southwest of England. So as a result, we got 257 serum samples. The eagle-eyed amongst you will notice that that is more than the 121 animals. And the reason for that is that in our long-term study, we were able to recapture individual animals several times during the period of the study. So the serum samples were subjected to a commercially available ELISA test. And we also carried out VNT for Delta and Omicron. Throat and nasal swabs were taken from animals. We had 189 of those. And they were subjected to e-gene for SARS-CoV-2 PCR. So the results were a little bit of a mixed bag. So what you can see in this table is the red dots indicate a positive reaction to one of the tests. Down here, each row represents an animal. These animals here, these top 6, these are all animals from our from our wild population, a wild sample population. This animal here was an animal that was wild but taken into a rehabilitation centre. And to sort of summarise the results, none of the ELISA positive results that we had, which you can see up here, had confirmatory VNT results, and so they didn't fulfil our criteria for confirmed positive status. We had 2 weekly VNT positive animals, these are the yellow dots here, for either Delta or Omicron, but they were not positive for both, and they weren't positive on the initial test and the retest. So they also did not fulfill our criteria. And so we have called these animals inconclusives. I'll return to that theme in a moment. But one animal was positive on VNT twice, both for Delta and Omicron, and so that does fulfill the criteria for confirmed positive status, and that was the animal that went into the rehab centre. None of the animals, none of the swabs proved to be PCR positive. So a little bit more about that confirmed positive case in the rehab centre. This was basically a cub suspected to be orphaned, its mother perhaps killed by collision with a car. The cub was found wandering around with its sibling in June 2021. It was taken back to the rehab centre where it was mixed with other cubs after it had proven negative on a TB test. So these rehabilitated cubs are always tested for TB. So it was negative for TB, mixed with other cubs. And then samples were taken in August 2021 for our study, so a swab and blood. But at the same time, it was TB tested again, and this time it went positive, and so it was euthanized. The other cubs that it was mixed with were, however, released because they all went negative on the TB test. But unfortunately, for whatever reason, no samples were taken for our study. So during the course of rehabilitation, the process involves minimal handling of the animals in order that they don't become too habituated to human presence. And of course, the million-dollar question here is, was this animal infected before or during the period of captivity? So the staff were all wearing PPE, there was minimal handling, as I've said, and they were regularly testing themselves before going to work, which will have limited the chances of exposure but not eliminated them. The cub, when it was first captured back in June, it was estimated to be about 14 weeks old. Now, badger cubs are raised underground, and for the first 8 to 9 weeks of their life, they spend entirely underground, and then they emerge at about 9 weeks or so. And they're weaned by anything between 12 and 14 weeks. So this animal would have been very dependent on its mother, and it would have had very limited contact with other badgers, because mothers defend their cubs against other badgers and isolate them in part of the set away from the rest of the group, typically. And very limited opportunity for exposure to humans, direct or indirect. So what's our interpretation of all this, and what's the broader context of our results? So we have this one confirmed positive. If I were a betting man, I would say that the balance of probability is probably on it being infected whilst it was in captivity. That seems the most likely situation. It's unclear what our inconclusive test results mean. We know very little about coronaviruses, other coronaviruses in badgers, so who knows what else is out there and potentially cross-reacting with the tests that we're using. And I'm very interested to see that there's a flash talk later on about a novel coronavirus in badgers, and I shall be talking to the presenter about this issue. So what we do know about SARS-CoV-2 in badgers, there was some ACE2 receptor mapping work that was carried out by colleagues in pathology and virology at APHA. And this showed that the distribution of ACE2 receptors in mink was pretty similar to what we see in badgers. So that would indicate that they're likely to be similarly perhaps susceptible. And then there is some evidence from a study in France showing antibodies to SARS-CoV-2 in wild mustelids, including badgers and pine mines. So what are the implications of all of this? Well, you'll remember that earlier on I said that most mustelids are relatively solitary for most of their lives. Well, badgers are the real exception to this rule, so they are not like other mustelids. In fact, they live in groups. Some of these groups can be quite large. So in our study, which has been running since the 1970s, in the heyday of the study, when we had lots of badgers in our population, we found some groups with numbers of animals totaling about 20 to 27. So these groups can be quite big. Most of the animals, they'll spend most of their social life underground with one another in a fairly confined environment. And there's lots of social interaction, social behaviour that brings them into contact with one another. But they're territorial, so each group defends a territory, but where the density of badgers is high, these territories are contiguous with one another. So across a landscape, you can have contiguous territories belonging to several different groups of badgers. And they can reach really quite high densities, so you can have in excess of 20 animals per square kilometre, for example. So they're very unlike other mustelids, and that might mean that transmission and persistence of a viral pathogen might be more sustainable in these populations. What about the opportunities for exposure to indirect or direct infection from humans, or even spill back into humans? Well, badgers are really well adapted to human-made environments, so you'll find them in lots of agricultural landscapes. You'll find them on the outskirts of Belfast. They're very well adapted to suburban environments as well. They're protected across the whole of the UK, but they are managed under licence. So they're managed in terms of trying to reduce their impact on development, so moving sets out of the way of railway developments, for example, and housing developments. They're also managed in terms of damage that they might cause to property by their burrowing, and they're also managed for TB control, both by culling and by vaccination. So there are some potential interfaces there. So I think when you bring all of this evidence together in a framework like the framework that we published in 2021 for trying to identify priority species to target for further work, then you can see that badgers are certainly worthy of further investigation. So I'll leave it there. Thanks very much for your attention, and I'm happy to take some questions.
Thanks, Des. So we have time for maybe one question.
Hi, I wondered, do you think it might be useful to test the indeterminate samples from the neutralisation assay on the pseudotype-based neutralisation assay?
On the what, sorry?
The pseudotype-based neutralisation assay that Grace Tyson described yesterday.
Right. I was really hoping that my explanation that I'm an ecologist at the beginning of this would, Fortunately, I have colleagues from the virology team who might be able to answer that better.
I think there's just a nod there, but yes.
The answer's yes. I mean, one thing just to say on those animals that we had inconclusive test results for, we did go back because we've got an archive of material of serum from these animals. And so we went back to previous captures and we looked at serum samples from previous captures and subsequent captures. I think there was only one instance where we had an animal that was ELISA positive on 2 different trapping events. All of the others were just positive on 1 event. But yes, I mean, I'd love to throw a whole bunch of other different approaches at this to try to get to the bottom of what's going on with those inconclusives.
Des, I think you've given someone else some work to do there. Okay, thanks, Des. Okay, and the next talk is from Kyle Perrins from UK Health Security Agency. Thank you, Kyle.
Thank you very much for having me here. I'm Kyle Perrins, as previously introduced. I'm a research scientist for the Virology and Pathogenesis group at UKHSA, and I'm just going to be going through research done by our group today. So first, a bit of background. So in 2021, there was data coming out of North America suggesting that white-tailed deer, as seen here, were able to act as SARS-CoV-2 reservoirs, with SARS-CoV-2 effectively transmitting and circulating in populations. This data, among others, included a seroprevalence study in Texas, which found positivity rates of 40% in white-tailed deer. Additionally, they also found the presence of neutralizing antibodies. Furthermore, there was also a confirmed white-tailed deer to white-tailed deer transmission in a captive deer facility in America. So this all paints the picture that SARS-CoV-2 is actively transmitting and circulating between white-tailed deer in North America. So this is, of course, very significant for North America, but for the UK context, why is it significant? If our deer in the UK were able to act as SARS-CoV-2 reservoirs, this could have a few implications. One, deer in the UK have a large distribution and range. There's a lot of deer species across the UK in a lot of different areas. So if a deer population was infected with SARS-CoV-2 and acting as a reservoir, there'd be a risk that it could spread all across the UK to other deer populations. Secondly, this deer SARS-CoV-2 reservoir may act as a source of new infections for other wildlife species, which may in turn lead to new opportunities for SARS-CoV-2 to evolve and transmit to other species and produce new novel variants. So as a result of this, the World Organization for Animal Health recommended monitoring of cervids to increase our understanding of the likelihood of domestic deer populations to act as SARS-CoV-2 reservoirs. So upon their recommendation, we set out to determine the exposure of UK deer populations to SARS-CoV-2. So a bit of context about the UK and its deer. So the UK has 6 deer species present, as seen here, varying sizes, and they're quite phylogenetically diverse between themselves and between white-tailed deer. So white-tailed deer is here in orange, and the UK deer species are here in blue. And as you can see, it's quite phylogenetically diverse. They also have different ecological and behavioral traits compared to white-tailed deer. So a quick mention before we go any further. Just before and during the COVID-19 pandemic, we had a separate serosurveillance study ongoing, and this was mainly focused on tick-borne encephalitis virus. So in this study, we used deer as sentinel species to evaluate the level of tick-borne encephalitis virus seropositivity in the UK. And this resulted in the detection of TBEV for the first time in the UK, as seen here. But more importantly and more relevant for our study here, the samples collected in this TBEV study were able to be used in our SARS-CoV-2 study and tested. So this significantly helped the speed at which we were able to evaluate the UK deer population for SARS-CoV-2 exposure. So our study aim was to conduct a serosurveillance study of UK deer populations and determine the exposure to SARS-CoV-2 and if it was circulating in the UK deer. So we determined there could be 3 possible outcomes for this. 1, there was no exposure of UK deer to SARS-CoV-2. 2, there was exposure, but there was limited or no transmission between deer individuals and deer populations. And 3, exposure, transmission, and circulation between deer populations in the UK. And of course, this would be the worst outcome. So to go over the methods. So firstly, we collected deer blood samples from routine culling and opportunistic sampling between January 2020 and May 2021. So as you can see here, a hunter has culled the deer and is using the syringe to put it into his chest cavity and extract about 50 ml of blood. So in total, we managed to collect 1,758 samples. Of these, 1,157 were females, 567 were male, 24 were unspecified. And there's the breakdown in species if you're interested. So once we collected and obtained all these samples, we then tested them with a Roche Alexis anti-SARS-CoV-2 N and S immunoassay. So this is a species-independent immunoassay, and it qualitatively detects SARS-CoV-2 antibodies against the N or S protein. And if there were any positive samples on the Roche assays, we then conducted a confirmatory surrogate virus neutralization test. And this assay was also used in North America, in the North American studies. And the assay detects neutralizing antibodies. So this is basically can help us discriminate whether we've detected cross-reactivity in the Roche test or actual infection, because actual infection is highly likely to produce neutralizing antibodies. So once we determined the methodology, we then could have a criteria for probable exposure. So 2 criteria for probable exposure, either one would have resulted in us assuming it was a positive result. So the first is that the sample was positive on the N and S immunoassay. And the second criteria that could have been possible was the sample was positive on either the N or S immunoassay and subsequently positive for neutralizing antibodies on the SVNT assay. So now that we've determined the criteria, I'm going to show you some results. So firstly, this is the sample distribution. As you can see, Majority of our samples were collected from England and Scotland, only 2 from Wales and 0 from Northern Ireland. And it also displays how the S-assay reactivity we detected is largely similar across the UK. But more importantly and more interestingly, here's the N and S immunoassay results. So you can see on the left there, this is the S-assay graph, and on the right, the N-assay graph. And here, the dotted line is the positive cutoff point determined by the manufacturer. So as you can see, the vast majority of samples are below the cutoff point, suggesting that they're negative. And there's only 54 samples above the cutoff point on the S-assay, and 9 samples above the cutoff point on the N-assay. Furthermore, 63% of the samples above the cutoff point on the S-assay were actually only marginally above the cutoff point. And, no one sample was positive for both N or S antibodies. So of these positive samples that we detected, 63 in total, we then went on to do the confirmatory SVNT assay. And as you can see pretty conclusively, none of them tested positive for neutralizing antibodies. So this allowed us to effectively conclude that none of our criteria for probable exposure had been met. No one sample was positive on both the N and S Roche immunoassay, and no one sample was positive on the N or S immunoassay and neutralizing antibodies. So this allowed us to basically conclude that there was no serological evidence of SARS-CoV-2 exposure, transmission, or circulation in UK deer populations between the study period. So of course this is interesting because our results differ quite starkly to those found in North America. And there's a few reasons that we think could be, but there's obviously probably a lot more, and it's probably multifactorial. So these are only just a few possibilities. We aren't quite sure why. But one reason that's probably highly likely is there is biological differences between UK deer species and white-tailed deer. That's pretty evident. Secondly, it's possible that UK deer have different ecological and behavioral traits. For example, maybe UK deer are less tame and more skittish than white-tailed deer in North America, so more likely to avoid human contact, thus decreasing the chance of human-deer interactions and possible SARS-CoV-2 transmission between humans to deer. But again, this is just a theory. Ooh. And thirdly, another possible factor is there's differences in human factors. So the population density and distribution across the UK is quite different compared to North America. The population density is much greater in the UK and tends to be less distributed, meaning there could be swathes of land where deer can roam relatively freely without encountering another human, whereas this may not be the case in North America as much. So, in conclusion, any further steps that we should take are we should do some further research and serosurveillance studies, particularly considering that this study was only conducted between January 2020 and May 2021, and of course there's been about a year and a half since then. So Omicron may have had a different effect on deer, may have been able to more readily transmit or infect deer. So that would be useful to investigate. Secondly, it would also be interesting to investigate the reason behind the N and S immunoassay cross-reactivity that we saw. This may be a deer-associated coronavirus that we don't yet know about, that may be similar to SARS-CoV-2 in relation to the N or S protein. So further research into that would be very interesting to investigate. And I'd like to thank everyone who contributed to this paper, and I'd like to thank especially the volunteers who collected the samples for us and culled all the deer. Thank you very much for listening, and I'm happy to take any questions.
Thank you, Kyle. We have time for a couple of questions. Maybe I can start. So you said that the plan is to continue some of the studying. The sampling, is it sort of routinely done, or is it now stopped? I mean, do you have some samples to go back to?
We have some samples. So at the moment, the sampling has stopped. We're still waiting for more funding to do some more sampling studies. So at the moment, we can't do anything, but I think that is a future aim and a possibility if we do get the funding to. If we're going to test it for TBEV, we may as well test for SARS-CoV-2 seropositivity as well. So I think that is a future possibility if we get the funding.
Nice talk, Kyle.
Thank you.
I was going to ask whether you've got any knowledge about surveillance for infection in deer farms or deer parks, where they might be more likely to be more heavily managed and come into contact with people more often.
So I do know, I don't want to preempt, but I did see in the study that there was exposure and confirmed cases of coronavirus SARS-CoV-2 in deer parks in Dublin, and I presume if we did any studies in the UK, we would also find that. But because our studies were mainly going off wild deer and piggybacking off the infrastructure of the tick-borne encephalitis virus collection study, I think we didn't actually have any captive or peri-urban deer in our study. So I think if we did investigate that, we might have a different result, but it'd be something to look into.
Thanks.
I guess this last question for me then. So any plans do you have any cell lines made from these different deer to see if you can do any in vitro experiments to look at the permissivity?
That's a good idea. I don't think we have any deer cell lines to our knowledge, but that would be a good and interesting study because it would be useful to confirm that the deer species that we have in the UK are susceptible to SARS-CoV-2. But I think theoretically they should be, as they all have the ACE2 receptors present. But yes, that's good.
Okay, thanks very much, Carl.
Thanks very much.
And just before coffee, we're going to move to our flash talk presentations. So our first speaker is Nazia from the Pirbright Institute.
Hi everyone, I'm going to talk to you about some of the work we've been doing to assess the species tropism of SARS-CoV-2 variants. So, as we know, in the past there have been outbreaks of other zoonotic viruses related to SARS-2 that have transmitted to humans via intermediate hosts. But the thing that all of these viruses have in common is that they originate from bats, and it's widely accepted that SARS-2 also originated from bats and then transmitted to humans either directly or via a yet unknown intermediate host. So what we've been trying to do is understand how SARS-CoV-2 and the subsequent variants have been able to maybe infect wildlife or livestock to assess the risk to these animals, but also to understand new animal reservoirs that could act as a means of sustained transmission, as has been described by other speakers for mink and white-tailed deer. So to do this, we're using low containment assays using a lentiviral pseudotyping system that was described by Grace yesterday, and also using cell-to-cell fusion-based assays. So we've been screening 22 different mammalian ACE2 species against the Wuhan variant of SARS-2 and of SARS-CoV-1. And as you can see, all these viruses show broad tropism for, all these mammalian ACE2 have broad tropism for these viruses, but we see some clear restrictions with rat ACE2, some of the bird species, and some of our bat species. So given the emergence of SARS-2 variants, we wanted to see if there was a shift in receptor tropism with the emergence, focusing on those where we saw differences in our original screen. So what we saw then actually was that the rat, mouse, and ferret ACE2 was then better used by the alpha, delta, and Omicron variants. And we were able to pinpoint this to key mutations within the receptor binding domain of the spike. So for example, the alpha variant has a single mutation in the RBD at the position 501. And we can see that this mutation alone was able to overcome the restriction we were seeing in these species. So of course, this doesn't necessarily mean that these animals are able to be infected by the virus, as Angela showed us yesterday. But what it does do is it gives us a tool to be able to quickly screen a large number of ACE2s to be able to understand which animals should be surveyed and looked at for potential virus spillover. And one of the sort of key animals that we're sort of now looking at further are the bats. So for the B1 strain of the virus, we saw that it didn't use horseshoe bat ACE2, which was surprising considering this was thought to be the reservoir for SARS-2, but it did use fruit bat and little brown bat ACE2. But then we see with BA1, this then shifted and the virus was able to use horseshoe bat ACE2, but there was a reduction in the usage of the other two. And we also corroborated this with some live virus experiments where we infected cells overexpressing these receptors with BA1 or B1. So again, we wanted to have a look at what mutations were important that could be explaining this change that we saw. So we generated mutants in the spike protein that had individual mutations that we thought may have been contributing to this. And we saw that the 493 mutation in combination with the 417 and 484 mutation BA1 was able to explain this difference that we saw. And finally, we're now expanding our ACE2 library to look at other wildlife species. One of those is being monkeys. So we're using a slightly different approach here, where we're looking at surface expression of ACE2 bound to RBD. And what we can see is for wild type and BA4 and 5, that these RBDs bind better to old world monkey ACE2s, but not to new world monkey ACE2s. And again, we think we can explain this by key residues within the RBD ACE2 binding interface, where positions 41 and 42 have been predicted to be regions of restriction to binding of SARS2 to these ACE2s. And we're now also expanding our analysis to look not only at SARS2 and their variants, but also other alpha and beta coronaviruses. So thank you.
Thanks, Naz. Time for one quick question.
Again, from a position of ignorance, I think you're normalising to the human ACE2, aren't you? So is it possible that if a virus, one of these, adapts to another species, that it just becomes rubbish for the human? I mean, that's what we might hope for.
Yeah, I mean, it's possible. So I think for some of the later variants, at least, what we're doing is now just trying to look at sort of the raw value rather than normalising it to be able to really understand how that virus is using that ACE2, because it's possible that if we're comparing it to something where human ACE2 is already rubbish, then actually you're not going to see that difference as well.
Okay, thank you, Naz. So the next speaker is Gianpierro Zamperin. He's going to talk about vouchers.
Good morning to all. Thank you for the organizer for inviting me here. I am Gianpierro. I work at the Istituto Zooprofilattico Sperimentale delle Venezie, very long name, in Italy. I am a bioinformatician and I work on NGS data, mostly on viruses and their host. The work I'm going to present is the discovery of a new putative genus in the coronavirus, in the Coronaviridae family, found in a Eurasian badger, which were already introduced a couple of presentations ago. So thank you for ruining my surprise. No, I'm joking. This discovery was done during a program of passive surveillance in Italy. This program has two different aims. One was clearly related to the COVID-19 pandemics, so was to exclude the establishment of a reservoir of SARS-CoV-2 in the wildlife. The other aim was to discover if there was some kind of local diversity about the coronaviruses. The program used more than 100 dead Eurasian badgers found in the Italian territory. From all these dead animals, we take about 300 of samples divided between intestines and lungs. We screened all these samples with a pan-coronavirus PCR. 2 of these samples turned out to be positive, so we did Sanger sequencing followed by an initial phylogenetic analysis, and from this, first of all, we confirmed that that was a coronavirus, and then this initial analysis showed us that that could be a potential novel species of in the Coronaviridae family. So we tried NGS sequencing on these two samples. Only in the intestine sample we were successful, mainly because both the samples was of very low quality. In fact, the intestine has a real number of 2.3. The other one was even lower. However, we are successfully in library production. We are successfully in the sequences. We produce with the NovaSeq about 500 millions of pair-end reads after applying different filters, including a quality filter, a filter on the host reads, and a taxonomical filter. We get 40,000 pair-end reads, which were of high quality and were clearly belonging or with the potential to belong to the Coronaviridae family. All these pair-end reads were de novo assembled. We get 3 different countings. The longest one represented the polyprotein, not the complete one, since it was lacking the initial 4,000 bases. The other counting represented a partial spike, and the third counting assembled represented something that could be potentially the nucleocapsid. So the total length of the 3 countings combined represent basically half of the estimated size of a Coronaviridae genome, and we checked in the ICTV guidelines which were the requirements to assess if this could be represent a novel species. We were very lucky in this, since all the 7 proteins here encircled by red circle were the requirement to assess new genomes, and were all present in the polyprotein assembled, and all of them has less than 46% of identity with the other known Coronaviridae proteins, and this was enough for the ICTV guidelines to assess a new genomes. So we we define it a new genus in the Coronaviridae family, and with a lot of originality. We call it epsilon coronavirus, and basically the take-home message of this work and of this presentation is that in the wildlife there is some degree of diversity which is still unknown, and revealing this diversity would be very important for the future, not only from an ecology and evolutionary point of view, but also to be better prepared for future pandemics that could arise in the future. Okay, so thank you for your attention.
Thank you, Gianpierro. Time for one question. Marike.
Thank you. Very interesting. I wanted to know, do you still have sample material left?
No, we have only the material enough for 2 different libraries and one final Sanger sequencing that we did on the non-structural protein 15, since it was not only very different from a similarity point of view from the other known one, but also it was longer. So to exclude some bioinformatician mistakes or whatever else, we designed a couple of primers in the assembled sequence. We sequenced it with the last 4 I don't remember the quantity from the last left samples, and the Sanger confirmed 100, totally confirmed the de novo assembly. But we have no samples left, so we cannot perform any analysis, and we are hoping that other that the surveillance program could be produce other samples to check the result in different animals, basically.
You answered exactly what I was going to ask. If you could then just follow up what the bioinformatics was telling you, which you did. So are you then going to develop primers to do further screening for specifically that virus?
It is a thing that we're going to develop, since this work is already published as a simple discovery of a new genus, but we have in preparation a wider paper about the surveillance, in which we will discuss about developing new diagnostic tools for better uncover the diversity that could be potentially present.
Okay, excellent. Thank you.
Thank you.
Thank you, Gianpierro. Now, final speaker before coffee is Nicola Fleckler from UCD.
Good morning. So I'm briefly going to tell you about some serosurveillance work we've been doing on a city population of fallow deer in Dublin. So this population lives in the Phoenix Park in Dublin. It's about 1,750-acre park in Dublin city centre. It's the biggest city park in Europe. You can see the train station there to the left of the image, and that whole green area is the park. So the animals, there's about 650 fallow deer in the park. They've been there since 1662 when they were introduced. About 80 to 85% of them in any year are individually identified with coloured ear tags, so they can be monitored behaviourally, and they are a very well-studied population. The Office of Public Works manages them, and they're managed culls yearly with around 90 animals culled per year. And the School of Biology at UCD has really studied their behaviour and interactions with humans very well. As you can see here, the animals have significant contact with humans. Here's a guy giving a deer a drink of Coke, which it seems to love. But they literally, the School of Biology know how many times the animals have begged from humans in their whole life. So they are all ranked according to their begging status within the entire population. Okay, so we used the GenScript serum neutralization test, and we have 3 dates. There are 6 cull dates altogether, 2 in November 2020, 2 in November 21, and again in February 22 there were 2 cull dates. So you can see that in November 2020, when B1 and alpha were the main variants circulating in Ireland in humans, the animals were pretty flat negative. In November 2021, when Delta was the major circulating variant, you can see it's starting to come up a little bit. And then in February 2022, when Omicron, so BA1 at the beginning of the month, and then BA2 and BA3 emerging, 57% of the animals were actually above the cutoff point in this surrogate neutralization test. All of the retropharyngeal lymph nodes, nasopharyngeal mucosa, tonsils and cecal content were all SARS-CoV-2 PCR negative using E and N1 gene targets. And this is just showing you the begging data for the 3 dates. So if you look at February 2022, the animals, all of the seropositives are ranked as occasional or consistent beggars taking food from humans. Now, you'll notice there's the rare animals and the avoiders are actually not there because they're also very good at avoiding getting shot. So that's the caveat with our data there. Sorry. So we next, we selected some of the 4 of the highest titer sera from 2022, and one of the sera from 2021 that was just on the border of the 30% cutoff for the virus neutralization test assay. So you can see the first one in gray does not neutralize SARS-CoV-2 pseudoviruses. So they were bearing alpha, delta, Omicron BA1, and BA2 spike proteins. But the 4 seropositive data on the virus neutralization test very effectively neutralized all of the the variants in the pseudovirus assay, which was quite surprising to us. So next we looked at fully infectious virus. And on the top we looked at an Italian isolate from 2020. So this would be sort of representative of the ancestral virus. And then we compared a clinical isolate of BA1. So you can see now the data is a little more nuanced. Some of the sera were able to neutralize the ancestral virus, whereas others were not. But all of them were able to neutralize Omicron BA1. Even the serum sample that was taken when Delta was the major circulating variant in Ireland. And so the next thing we wanted to do, because we didn't have any PCR positives, we generated precision-cut lung slices and tracheal explants cultured as air-liquid interfaces from freshly culled animals. So once the animals were culled, we got the lungs and tracheas into ice-cold PBS, got them back to the lab, generated the precision-cut lung slices and the tracheal explants, and then we challenged them with the ancestral fully infectious virus or the Omicron BA1. And 72 hours post-infection looked at immunohistochemistry for SARS-CoV-2 spike protein. So you can see in the tracheal explants, the pseudostratified epithelium is immunopositive for SARS-CoV-2. This is only with BA1. The ancestral virus was unable to infect these tissue slices, even though the titer was slightly higher actually than the BA1. And the precision-cut lung slices, we got very nice immunoreactivity in type 2 pneumocytes, mainly in the lungs. So these have been blindly scored by both a human pathologist and a veterinary pathologist, and they're in agreement about the results. So only BA1 was able to infect these. So I think this really, to our knowledge, it's the first description of SARS-CoV-2 seropositivity in a European population of deer. And I think it really underscores the importance of continued surveillance as new variants emerge. So I just want to briefly thank my research group. They're absolutely amazing. This is an unfunded project, and they all took time out of their studies to take part in this. Particularly, I want to thank Dr. Kevin Purvis, whose idea this whole project was, and also to Simone Cuti's lab in the Laboratory of Wildlife Ecology and Behaviour at UCD, who's been doing all the behavioural work on these animals for many years. Thank you.
So we have time for just one quick question before coffee. Sharon.
Thanks, Nicola. I've actually got a question about the behaviour rather than the virology, strangely.
So, I'm not a behaviourist.
No, that's fine. But it's just, it would be curious to know why they saw the increased begging over that period of time. Was it because there was more people, less people in the park? Were they doing something different? So other than feeding them Coke, was there any other, changes in that contact
most
that allowed
definitely
for transmission?
There was considerably increased contact because we were in lockdown. So people had nowhere to go. So all of the people in the city, they were allowed to walk 5 kilometres from their home, so they went to the Phoenix Park and fed the deer. And I mean, over the last 10 years, there's been increased contact with these animals. And Dr. Cutie's group kind of attributes this to social media and, how cute the animals are, and you can feed them Coke and things like that,
Great. Thanks, Nicola. Okay, coffee. Can you please come back here just before 11:15 for the next session? Thank you.